Yeah, hello everyone. Thank you very much for having me. So we heard a little bit today already about CytoNet, which is a foundation model for the human cerebral cortex. And in this talk, I'm going to shed a bit of light on what CytoNet actually is, how it works, and what it can do, with a particular focus on its application in big brain, but also beyond. So let's start with a brief summary of what CytoNet is. CytoNet is a deep neural network which takes microscopic image patches, as we see here, at around 1 micrometer resolution, extracted from the cortex, and it encodes these images into feature representations. Think some kind of vector representation that encodes what kind of cytoarchitecture we see in the respective image. Why would we want to do this? Well, it turns out these representations are a very, very powerful foundation for doing downstream analysis tasks, which means that after having a model that can produce these features, we are able to solve tasks like brain area classification, cortical layer segmentation, parameter estimation, and data-driven mapping. In this sense, CytoNet is a foundation model, and before I go into detail like how the training works and how we can actually apply it, let's have a look at what we actually mean when we talk about a foundation model. So according to a quite well-accepted definition, a foundation model is a model, a deep neural network, that is trained on a broad amount of data, generally using self-supervision, and we will come back to that in a second, with the goal to be adapted to a wide range of tasks, which is basically a shift away from what we have been doing over the last few decades, from having first in the, let's call it the machine learning age, doing feature engineering, thinking about what we need for a specific task, then towards deep learning, where the feature engineering is actually done by the model itself, but we still have expert models for different kinds of tasks, and we're now moving into the foundation model age, where we are basically training generalist models, which once trained can be applied to a wide range of different tasks. So one could say that we are aiming for one model to rule them all, but there's a little star there because, as you might have noticed, we have multiple foundation models around. I mean, we heard about one or two already here. This means that we are not yet there, and we might never get there, that we only have one model for all kinds of different stuff, but we are basically lifting the boundary a little bit from to at least on a domain level have less models which are more powerful. And here are some examples which are like generalist models, for example, for text, images, and there you see even within one domain, let's say text, we have multiple competitors, which is not necessarily a bad thing. So one important thing of these foundation models is self-supervised learning, which is the way that we make these models actually learn the things they should learn to be useful at the end of the day. And the idea here is that we train the model using the inherent structure of the data, meaning that we don't provide an explicit supervision signal through labels or so on, but exploit the data itself. How does this look like? Here are some examples. So for example, we might ask the model to solve jigsaw puzzles, which are completely free. We can chop up any image and then ask the model to solve that. And in text, can do just stuff like this, like filling in the gaps. So the point here is that we are learning strong representations as a basis for our tasks without using labels. This is important. So we are basically only limited by data and compute, which means that while developing these models, we are kind of shifting away from gathering labels and so on, but instead thinking about, okay, how can we trick our model into learning what we want? So how do we do this in CytoNet? We use an approach that we developed called contrastive learning from spatial proximity, where the goal is basically make the model map images which we deem similar in some way or form to be encoded similar. And our approach here is that we say close means similar. So the closer two locations are in the brain, we say these should be encoded more similar. Of course, you might have objections here because this is definitely not true in a lot of cases, but it just has to work well enough so the model picks up like the overall idea, and it turns out that the details are not actually that important. But to make this a little bit better understandable, let's have a look at an example. Suppose we have two brain scans from two different brains, but from approximately the same location, and we extract some image patches. And we see that the blue and the orange one are, even though they are in different brains, they are quite close to each other, while the purple one is a little bit further away. So what we say now is that blue and orange should be encoded in a similar way, while the purple one should be encoded in a different way. Now, the technical detail is that we can't exactly compute distances between brains, so we actually don't measure the distance in the sections, but we do this in a reference space, in our case in the MNI colon 27 space. But the actual space is actually not really that important. You can essentially use what's most convenient. And now we have everything that we need for our model. So we can extract high-resolution image patches. We can pass them through a neural network, get feature encodings, which are these little boxes, then impose the similarity that we just computed based on distance, and then optimize the model to basically replicate the similarity relationship. This is the loss that we developed, the objective function, the training function, for those of you who are interested, that we developed for this, which is basically an info-NCE loss, quite standard loss for contrastive learning, with the addition that the similarity that we impose here is actually derived from the spatial proximity of the image patches, and we do a pairwise comparison across all different kinds of image patches that we have in our training batch, which is an approximation of comparing all patches that we have within the brain. So this is the method, but the method alone is not enough. We also need to train this. The way that we train this is we take data from 10 brains, which are parts of the Jülich-Düsseldorf brain collections, about 4,500 sections, which is about 11 terabyte, of which, after we sample, in this case here, 1 million image patches, this is about 4 terabyte, which is then processed multiple times by the model. So one training actually accounts to about 600 terabyte of data seen by the model at some point, and corresponding locations in colon 27, for example. And the image patches that we use here is about 2K by 2K image patches at 2 micrometer resolution. The architecture is very flexible. We have found that ResNet 50 and Vision Transformer hybrids work quite well, and that larger models tend to work better and more data works better. So actually this is just a current snapshot, but we are actually working on at the moment of scaling this up. This current configuration here takes about 28 hours on 64 GPUs, which is about 65 GPU days. So let's have a look at what we can actually do with this model. We heard about this already. Brain mapping is a very, very prominent example, and I apologize that I can't go into too much detail on each of the applications. I just want to give you an overview. Where, in this case here, we basically train the model to, based on patches like this, we ask it which brain area we see. And it turns out that the model is actually quite well capable of doing this. We achieve a macro F1 score of about 75% and a top 3 accuracy of 95%. So it's actually very, very well capable of actually doing this task quite well. And very recent results, so in the sense of doing it in the train on the way here, results also indicate that this also works for subcortical areas without any specific pre-training for this. We can also do layer segmentation using the data set that we also saw this morning in Katrin's talk, where we basically asked the model to do a segmentation task this time. And here what's very interesting is that using these representations as a basis, we are very, very label efficient. We can go down to, this is the results that we see here, up to 7 annotated patches to train this model to a reasonable degree. And those of you who have done this kind of annotation before will appreciate that you actually don't need to do that much to get reasonable results. We can do what is here phrased as feature prediction, so which is kind of a fuzzy term, but think about if you have some measure that you want to predict across the brain, let's say the cell density in a specific layer, we actually find that CytoNet can predict these quite reliable, which could, for example, be used for making whole brain maps of a specific layer density. And this actually works, what we compared here, better than, for example, using the popular intensity profiles that you can simply extract from the images. Complementing the approach of brain mapping, so brain area classification, we also find that these features lend themselves nicely to data-driven discovery of brain areas. So assume that we don't know yet in a region whether this should be subdivided or not. We actually find that simple clustering, maybe even a little bit more sophisticated approaches, can help us to recover subdivisions in brain areas. And what we did here, for example, is that we took an area which we know had 2 subdivisions and saw and checked whether CytoNet can recover the subdivision, and it actually does this with quite high accuracy, which is quite nice. In a similar direction, we can also use these features to do some kind of variability analysis. We see this here in a kind of embedding plot, where we see that actually the features that the model learns fan out with respect to the brain they belong to, which is interesting because the actual objective does not incentivize this. So this actually tells us that at least according to the model, whether we believe that or not, there is a very, very significant difference between the brains. We just have to tingle out what this is. And there are also some more applications. So we heard already in the talk by Ollie that we can use CytoNet for the quantification of generated images. It also provides the backbone for our interactive BigBrain mapping projects, where it's basically like the foundation for the actual models that are trained there. And we can also do stuff like model introspection, checking what does the model attend to when it solves a specific task, for example. This was a very, very high-level overview, which brings me to the last slide, asking what comes next. So I already mentioned that we want to scale up. So we want to try larger architectures and data. And for this, we are very happy that this project was selected as one of the pioneer projects for the new Jupyter Exascale supercomputer that has recently been commissioned in Jülich, where we are currently seeing how well we can scale up our workflows. And I'm quite happy to report that we can reasonably manage up to 12,000 GPUs for these trainings, which is quite cool, I think. We want to work on multimodal extensions, so we are currently investigating how well this works for PLI data and text data. As I mentioned, we are doing subcortical extension to subcortical data, and of course we are working actively towards bringing this actually out for people to use it in the terms of data publications and tools. And with this, I'm finished. I hopefully not too much over time. Would like to thank all my collaborators and people who contributed to this work, and I'm very, very happy to take your questions.